Okay, so I'm not going to talk about biobanking, so apologies for that straight away. I'm going to talk about cell biology, so it might form a nice segue into tomorrow's talks, I think, a little bit, and also functional genomics in particular. So let me give you an introduction to the company. So Horizon, a translational genomics company, we were founded in around 2007 with the explicit goal of generating precisely genetically defined cell lines for the research community. So these are cell lines which we will define in accordance to a disease, for example, to provide reagents that we can use throughout the community and also internally to model diseases in a more accurate and precise way. Since those early days, the company's grown a lot. We've expanded into lots of different areas of drug discovery, even into bioproduction and diagnostics. But the heart of the company is still in the generation of these isogenic cell lines, as we call them, because they're so precisely defined. And we've built products on that basis throughout the last 7 or 8 years. But the remit for the company has also been sort of bolstered by some really transformative technologies. The huge burgeoning amounts of genetic data that's available now has really improved what we've been able to do with the generation of our cell lines. But there's been a fairly clear remit also from studying the economics of drug discovery, where big blockbuster models have sort of been roundly assumed to have failed, really, and not delivered the sorts of products and the sorts of medicines to patients that we were really hoping for. So there's a real strong emphasis now on trying to reduce costs, and part of that is being able to model disease more accurately, more precisely, and reagents that are hopefully generated by Horizon are going to contribute to that whole process. So this is where Horizon comes in. This is where functional genomics as a field really comes in, is really trying to understand this now enormous quantities of genetic data and make some sense of it and make some contributions to improve research opportunities. So today, as I mentioned, we've got lots of areas of interest across Horizon, from early stage of drug discovery, patient stratification, all the way through to diagnostics. But I just want to focus on two areas, looking at some of the more recent developments in our cell line engineering program and products that we have available now, and then a recent launch of a functional genomics screening program, which I think is really exciting. So to give a background to this sort of cell biology that we're doing, this sort of genetic engineering has really improved over the past few years. Horizon was founded using a proprietary technology, rAAV, but now over the past 2 years, there's been this huge explosion in CRISPR-Cas9 technology. And this is a new technology platform that allows you to very rapidly and quite precisely genetically engineer mammalian cell lines. So it's provided really transformative. I expect people to consider this to be, the discovery of CRISPR to be as important as the discovery of PCR over the next 10 or 15 years. It's really enormous. In fact, almost touted as a Nobel Prize last week, so almost made it that far in 2 years, which is pretty impressive. We, as a company, are very agnostic about the technology we use, so we'll use any approach that we think is the most appropriate. rAAV has a lot of nuanced advantages in terms of precision, but it is slower. So we still are doing a lot of work in CRISPR now, and I'll try and explain a little bit about that technology on the next few slides. But overall, the principle is that we can genetically engineer these cell lines to generate a disease simulating reagent, including point mutations, full knockouts, any other genetic alteration that you can think of, and deliver that as a product. So some of the sorts of things we'll be looking at, we generate a lot of knockout cell lines. This collection's growing really, really rapidly. So we do this in a variety of cell line backgrounds. And these will be generated already. Ones are available off the shelf as a product, but we do a lot of custom work as well for more complex projects. Knock-in cell lines, these obviously extremely valuable as well. This is fed significantly into our diagnostic reagent production, and being able to open up new areas of new studies for us as well. The important point to make is that knockout cell lines can be homozygous, or in some cases actually it's more valuable to have a heterozygous knock-in to look a little bit more at some subtler aspects. But also a really exciting area is the availability of some new reporter cell lines. So what this new technology has been able to let us do is to start making reporter cell lines for new assays that rely only on the really endogenous events going on in the cell. So we're not worrying about ectopically delivered reporter cassettes anymore. Actually, we're looking at the protein stability or the transcriptional response of the endogenous loci, because we can go into that cell line and introduce this reporter cassette directly where we want it in the genome. I'll talk a little bit about that in a few slides' time. So just a few examples. This is really talking about CRISPR-Cas9. I recognize this might not be a familiar thing for everybody here, but to give a sort of brief overview, CRISPR is a nuclease sequence-specific directed enzyme which introduces double-strand breaks at relatively precisely defined loci. And that has been really transformative. But the simplest paradigm that you can use CRISPR-Cas9 technology in is to introduce a double-strand break at a defined loci and then rely on the endogenous repair mechanisms in the cell line to repair that loci. Those repair mechanisms are inherently inaccurate, and what you frequently get is an out-of-frame mutation, and consequently that's a knockout. If you introduce that double-strand break early enough in the exon structure, then the likelihood is that you'll generate a knockout. But there's some stochastic elements to it too, so there is some automation, some screening requirements that we need to go through to make the most of this. But some examples of a panel of cell lines where we've knocked out specific genes and, of course, lost expression. This is actually completed using a haploid cell line model that we have. So the advantage of a haploid cell line is, of course, that you only have one allele to knock out, so that makes the process that much quicker and can potentially introduce some new models for study. So we acquired a company called Genomics a few years ago, or maybe last year, and they have developed the KBM7 haploid model, which was a blood lineage haploid mammalian human cell line. Changed it essentially using CRISPR technology into a fully haploid cell line, so removing some of the duplications in the genome, and also adapted it to be an adherent cell line, which is sometimes more convenient for assay in certain aspects of biology. But this collection is really growing rapidly, about 100 cell lines a month on the knockout collection. We clustered it into different functional collections in kinases and epigenetics, and this is actually a really, really valuable reagent, both within Horizon, but also hopefully to other research community as well, because this actually feeds directly into screening opportunities, panels of cell lines which you can use directly for studies to define certain criteria in different disease paradigms. The CRISPR-Cas9 technology does work well also in more complex cell lines, in cancer cell lines. Here's an example of A375 cells, which are a hypertriploid cell line. We are also able to engineer these cell lines to generate a homozygous knockout cell line. As I mentioned, there are stochastic elements, and this is really where the automation comes in. We have a Hamilton Microlab star that we use to essentially allow us to go through a far higher number of clones to get to where we really want to go to, because we need to screen through sometimes hundreds or thousands of individual clonal cell lines to find the cell line which has the correct genotype that we're interested in. This was a good example. This took around 96 clones. This was quite easy, and we were able to generate, once we'd identified them and validated them sequencing and restriction enzyme digestion, we found 3 clones with the desired homozygous knockout in this particular instance. So the reporter cell lines are also really exciting for us. We've been working with Promega to use their reagents. In particular, we use a HaloTag and NanoLook. So the HaloTag is a really adaptable system. It allows you to introduce this cassette, which you can then essentially click on different moieties, a fluorescent or an affinity-based component that you then can tailor to your downstream needs in assay and research development. The NanoLook is essentially a miniaturized luciferase reagent, and we can use this again, as I mentioned, bring that in specifically into the loci of interest, and that can either be as a transcriptional reporter, or we can also introduce that as a fusion with a protein to look at protein stability. We have a number of reagents already available off the shelf, and again, as I said, the collection is growing, and there's a lot of custom cell line generation opportunities here as well. So then I want to give some examples of how we're using these tools, and one good sort of case study is really where we look at what we're doing in functional genomic screening. So Horizon have been doing this sort of screening for a few years now. We're particularly interested in synthetic lethality, which is about understanding different therapeutic opportunities in oncology and gene-gene interaction. And we were doing that for a long time using siRNA in an arrayed system. So again, we were then using our Hamilton microlab star to generate siRNA reagent plates and look through screening in a relatively high throughput. We recently also introduced a haploid system, which is a gene trap system that is a complete knockout, whole genome approach to looking at patient stratification by knockout of a given gene and response in the haploid cell line panel. But what we're actually most excited about, I think, at the moment is the advent of the CRISPR-Cas9 screening program that we've just launched quite recently. And this has got some really novel opportunities, I think, in many areas of biology, not just oncology. So now I can give a little bit more information about what CRISPR-Cas9, the system, really looks like. So it's an endonuclease originally discovered to provide, it was discovered as a bacterial system providing innate immunity in bacterial cells, but actually it's been repurposed to be really, really valuable in mammalian biology as well in research. And what it really is a nuclease which is directed to a particular loci with a short guide sequence. So this is an RNA component, it's a ribonucleoprotein. And the nuclease can either be a dual-acting nuclease on double strands, or you can adapt this to different variants of the Cas9 system as well. But what it really ends up doing is generating, as I mentioned, a double-strand break. You end up with an indel mutation in many instances, and that gives you this frame shift and consequent knockout of the gene. And what it gives us as a technology improvement is actually really to compare it to RNAi, where a lot of the shRNA screening infrastructure has been sort of hijacked by CRISPR-Cas9 to a certain extent. But CRISPR-Cas9 gives the opportunity for much lower off-target effects. This is an emerging field. There's still a lot to learn, but it looks like it's quite likely to provide that advantage. And we also get potentially far more robust phenotypes than you would do with just expression depletion, because we're getting complete knockout. Now, that might not always simulate as precisely your expected drug interaction, but it nevertheless provides a greater window for analysis so that you would hopefully be able to look for finding the greatest phenotypic change in any given instance. So how does our new screening offering work? Well, it works in a pool-based system, so much like the shRNA screening. So what we're looking at here is, rather than in an engineering context where we engineering one gene out in one cell line, we're looking at hundreds or thousands of knockouts in a single individual experiment. And we do that by defining a library up front, a pooled library, which uses hundreds or thousands of individual guide sequences, all targeting different genes. We will design that library for you. We've got a lot of growing kind of experience and expertise in designing the right library, but we also have a number of off-the-shelf libraries, so where we will target a collection of genes in an apoptosis pathway or cell surface, or a number of different available interesting target collections. And included in this is a whole genome library. So that targets around 19,000 genes in each instance, with 6 of these individual unique guide sequences in every case, so a little bit similar to multiple hairpin screening in shRNA. The program then is based around generating a lentiviral preparation from that, transducing cells in one pot of tissue culture reagent, and then introducing a screen assay condition, addition of a drug, a panel of cell lines, a certain growth condition, and looking to find the resultant phenotype using next-generation sequencing. So we'll be able to characterize quite precisely, quantitatively describe the population of cells at any given point during the screening process. So in the simplest example of this, you would be looking at finding essential genes. You'd be looking to identify a gene that was, when knocked out, killed the cells, and you say that in a longitudinal study, for example, at the beginning of the screen, I've got a certain number of these guides present. At the end of the screen, I've got far fewer. Therefore, that gene is classified as having a negative response in that particular experiment. So I recognize that's quite a brief overview, and there's quite a complicated process, but I want to take a bit of time to show you some of the data that we've been able to generate from this program as proof of principle, and to show you the sorts of things that we've been learning. So this is a whole genome screen looking at A375 cells, which are a BRAF mutant cell line, that are sensitive to vemurafenib, which is a BRAF inhibitor. And what we were looking for in this instance is to find genes that, when knocked out, confer resistance to this compound. And there are a number of known instances of resistance generation in tumours, some of which have been found in patients, but some have also found it in other functional genomic screening exercises. What we were able to show is plotted over here. On the y-axis, we're looking at p-values, at the significance of the effect, and on the x-axis, we're looking at essentially an enrichment score, a log-fold enrichment of that particular, in this case, gene-level effect. And what we identified is a number of hits shown here. All of these are enriched, so these confer resistance of vemurafenib. All of them have been identified previously, with the exception of a few that we identified in addition to extant screening operations. So 2 of these were found in RNAi screens, 6 were found early last year by a screen done in Feng Zhang's laboratory over in Boston. And we were able to identify all of those really very easily with this screening technology once we got it up and running, in addition to multiple other components that were found in shared resistance mechanisms against vemurafenib. So this is a really clear validation of the protocol at Horizon, but actually I think more excitingly, this really validates the technology itself, because this is a screen which was done with a library which was significantly different to that which has been used before in a completely different laboratory on different continents, and yet we're getting the same screen, and this is almost the same data rather. And this is almost unprecedented, I would say, in RNAi screening, and so it really does open up some new opportunities. Over here is just some more detail on the data, just to show you the enrichment score of individual guides, so that's individual reagents in this experiment, to show you that actually the enrichment across the experiment was in the orders of 1,000 and 2,000-fold. This is on a log2 scale, so really, really significant enrichment. So this is a kind of an ideal experiment. It's not always this easy. This is looking at enrichment. We're also really interested in looking at sensitivity, so that's where you're looking at 4 minutes, thank you. That's where we're looking at dropout of genes as a result of the effect in a given condition. We have also been able to do this. I'll skip through this quite quickly. This is a glucose starvation screen, so we're looking for genes that, when knocked out, sensitize cells to glucose starvation. We were able to find a number of NADH dehydrogenase enzymes, exactly what we expected to find in this instance. So we have been able to do that as well, which is really good. But now we're looking at ways to optimize the system. And this is really quite exciting, because what we're looking at doing here is, really improving this sensitivity screening program. So looking to be able to generate more robust data from this sort of paradigm. And what we're doing here is using essential gene loss as a proxy to measure the technology. So we're saying if you're a successful screen and you knock out, say, a ribosomal gene that we know to be essential, it should be lost very rapidly from the population. And that's what's plotted here: loss of genes that are targeting ribosomal components across the duration of a screen. So we are able to detect that. This is a sort of random collection of genes. But most interestingly, what we've been able to do with this sort of frame is actually... okay, well, I'll just talk. It's not that big a room. What we've been able to do is actually find improvements to the system. So the old system here is shown in blue, and Horizon's new backbone here is actually shown in orange. And these are all showing dropouts of different essential cohorts of genes. So you'd expect to see these dropping out of the population, but in our system, what you're now finding is that dropout on a log2-fold scale then is upwards of 10-fold improvement in the actual quality of the screening data that we're getting. So that's really, really exciting for us. Just to finish up then, just how can we integrate these two strands of interesting cell line development and interesting new assay development? Here's a good example of what we want to be doing in the future, and we're looking for new partners to help us with this, using engineered reporter cell lines in a CRISPR screening paradigm. So looking at a reporter cell line, introducing a library of reagents, and then using a complex assay, flow cytometry, to deconvolute populations of responders and non-responders, and we can then deep sequence those populations. So that will really open up what we can learn about biology outside of the context of just looking at the viability of a cell line. Now we can look into really other, more exciting areas too. And finally, we also think that we can use the cell line panels that we're generating, the reagents, the knockout cell lines, for validation as well, and to confirm hits, for example, that we get in screens. And here's an example of that, where we're looking at 6-thioguanine resistance, again in haploid cell lines. In this treatment, this is a DNA damaging agent. genes that confer resistance to this drug found in the mismatch repair gene as they transition through mitotic arrest, irrespective of because of the drug. And we're identifying several mismatch repair genes here. And then over here, what we've got is a dose-response experiment conducted with engineered individual cell lines, so clonal cell lines that are deleted, in this case, of these two mismatch repair genes that we found as hits. And what you can see is a clear shift in the dose response, representing a resistance acquired by this knockout following the loss of these two mismatch repair genes. So that validates these genes found over here. Perhaps more excitingly, what it really shows you is that this difference, which is on the sort of half-log sort of scale, is robust but relatively modest, but actually the genetic screening is really, really powerful, and you find these hits actually quite easily in the genetic screen, telling you that actually maybe there's really new opportunities that we've never even thought about before by using this technology. So I think I just about made it. And I just want to finish by saying lots of other exciting things. We are just outside that door, so there's no excuse not to come and talk to us if there's anything of interest here or anything I haven't been able to cover. So if you've got time for any questions, I can try.